Hi there, Mr. Rowe here. Welcome to my presentation on the Endosymbiont Theory for A-level classes. This starts off with a story going back a very long time, and as with any good story, it obviously needs to have a hero. These heroes are the prokaryotic cells, which were the first cells to develop. Nobody knows exactly how they came about, but the leading theories so far are the lipid membranes were first of all formed, followed by information. This information was either encoded as proteins, which were able to self-assemble passing on the information, or possibly they were due to RNA, which was able to come together and then to pass on the information. We don't know exactly how it happened, but these cells were able to divide and replicate and pass the information from one generation to the next. So these were the first replication machines, which is what all life does, and they were the very first living cells. Things weren't exactly a bed of roses for these cells though, and there was lots of competition between them. You had cells of different sizes and different shapes that were all trying to have the most relative surviving to pass on their genes to the next generation. A process that was happening all the time is to obtain nutrition. Lots of the cells would engulf smaller cells by extending their cell membrane and taking them inside the cytoplasm, then digesting them with enzymes. This is a fairly similar process to what still happens with white blood cells at the moment. Next thing that happened is somebody got lucky, or a cell got lucky. One of these cells, in engulfing another cell, was able to take inside it a primitive mitochondrion. So that is a prokaryote that had managed to find out how to do aerobic respiration, so it was able to produce energy from oxygen and glucose in a really efficient way. For some reason, the mitochondrion was able to survive inside the cell instead of being digested in a normal way. As far as we know, this is a very rare event and only happened once. The cell which took the mitochondrion inside gained a huge evolutionary advantage over its competitors. It was able to produce energy far more efficiently, which meant it had more surviving offspring, and the genes for producing the mitochondria were passed on to the next generation, along with the cell's own genes. In what we think is a completely separate event, something very similar happened to produce the precursors of plants. A cell which probably already had mitochondria and could respire engulfed a photosynthesizing prokaryote. Again, it would have survived in what we think is a very unusual, probably one-time event. These cells were then able to carry out photosynthesis using these chloroplasts. Now, to finish with the evidence for this theory. First really strong piece of evidence is when you analyse the DNA present inside cells. The mitochondria and the chloroplasts both have small traces of their own residual DNA, which is different to the DNA present in the cell as a whole. If you look at them under microscope, they're around the same size as many prokaryotic cells, and they're a lot smaller than the eukaryotic cell they're inside of. They also contain their own ribosomes to make proteins. These ribosomes are slightly smaller, they're 70S instead of 80S. This shows that they come from a prokaryotic source rather than belong to the eukaryotic cell itself. And lastly, the double membrane that surrounds them provides evidence for this theory. The first membrane would come from the prokaryote cell membrane before it was engulfed, and the second membrane would be arising from when it was engulfed inside the first eukaryotic cell. Thank you very much for listening. I hope you found this useful, and feel free to watch the video again if you need to go over some of the information.